Hello, I'm Morgan. This is Danielle, and this is Arin. We are from Grand Valley State University, and our mentor is Dr. Jen Moore, who isn't here. And we are studying the spatial distribution and demography of eastern Massasauga rattlesnakes. So some background first. They are found in the Great Lakes region. So the blue squares that you see on the map are extant populations, and then the little purpley pink ones are ones that have been extirpated from those areas, which means they're wiped out. And extant means that they're here. So as you can see, around Michigan and the Great Lakes, and then they also extend to other areas as well. They prefer wetland habitat, but they do move upland later in the season. So as we move into July and August, we'll begin to probably have more captures in the uplands. They're the only venomous rattlesnake in Michigan, as I think Liz mentioned. And they eat small mammals, amphibians, and other snakes, and pretty much anything smaller than them that they come across. And they are proposed for federal listing under the U.S. Endangered Species Act, which makes them important for study. So we have 3 objectives, the first of which is Arin and my objective, which is to analyze movement patterns and habitat use across seasons and years using GIS, which is Geographic Information Systems, which she will be talking about later. And then Danielle's 2 objectives are to estimate population census size, density, survival rates, and population growth, and the genetic effect of population size, and she'll be using different software, which she will talk about later as well. So our methods of data collection. So marker capture data has been recorded from 2011 to 2016. So we have several years now of data, which is really nice. Good for writing. We use visual encounter surveys, which means we just go out and use tongs and pick snakes up and take them back to the lab in a bucket. Many of the other student researchers have come out and done that with us. We have cover objects, which are boards that we place in areas of dense vegetation out in the field where snakes might need areas to bask to find them there. And then funnel traps are basically made up of wire, like mesh wire, but they're found at the end of a drift fence, which is a fence made out of tarp with wooden posts connecting the tarp. So the goal is that as a snake runs into the fence, they will slither along the fence until they get to the end and go right into the traps. We also record snake locations with a GPS, as well as our tracks to know where we have been walking around the property. We mark snakes with a pit tag or a brand. A pit tag is like a microchip, and we insert that below the scales. And a brand is made using a cauterizing unit on yearling snakes, so ones that are too small for a pit tag. And then we determine the length of a snake by putting them in this squeeze box. So the first picture, thank you, Arin. So as you can see, the snake is being squished. So it's very harmless, but there's foam underneath the snake and plexiglass on top. And then this is Danielle in the picture drawing a line with an X-fin marker along the snake's backbone, and then we measure it with a measuring tape to monitor how much they're growing. And then we also weigh them, so mass, we sex them, male and female, in reproductive condition. So when a female has eggs, she's referred to as being gravid, so that's like being pregnant. And then we also collect genetic samples, which Danielle will use for her part.
So, just a side note that this slide is just a tiny bit inaccurate since Danielle found a new snake this morning. But from 2011 to 2016, we have had 551 captures of 219 unique individual snakes. But just focusing on this year, we have had 83 captures of 64 individuals, 34 of which were brand new individuals with no pit tags, 30 recaptures from previous years, 20 of which are adult males, 24 of which are adult females, and 8 of which are gravid, which Morgan explained means pregnant, and 20 yearlings, which means that they were just born last year. And as far as Morgan and I's part of the project, we are using GIS, which she mentioned stands for Geographic Information System. And that is basically a mapping software that allows us to present and manipulate data that we collect. And our goals within GIS are to create minimum convex polygons, which we use, which we have to have 3 or more snake capture points in order to create polygons to give us an estimate of their home range. We also measure the straight line distances between successive individual captures to get a sense of their site fidelity, and that basically means whether or not they are returning to the same habitats or if they're moving into new habitat types. We have and will continue to extract land cover data from each capture site to analyze the habitat use and different land cover use that the snakes are found in. And we are also examining yearly movement patterns using animation maps in GIS. And that is going to be incredibly useful for tracking where they start off in their hibernacula and where they move throughout the year, throughout the season, and whether or not they are still using the same hibernacula areas. And we're doing this all with the help of our wonderful GIS expert, AJ. And just that's a little interface of what GIS looks like. And this is an example of a minimum convex polygon. As you can see, there's a larger polygon. This represents one individual male snake. And the two smaller polygons are representative of one female snake. So males tend to have a little bit of a larger home range, while the females have smaller ones. Every single green dot that you see is a snake capture point. Some of them are individual, or some of them are recaptures, but you can see that on the polygons. And the colors, the pixelated colors in the background are the different land cover use. So one issue that we have been running into is that our data isn't exactly lining up with the land cover use. So this blue right here is Brewster Lake. And as you can see, there are 4 dots. We did not find 4 snakes in the middle of Brewster Lake. So we will have to go back and analyze the actual data and see where we found it right here, probably in some nice wetland area. But other than that, that is a basic idea of what we will be doing in GIS.
Oh, no, I didn't. Okay, so I don't have a lot of time to get into a whole lot of detail on my master's project, so I'll give the very brief version. Using the capture-recapture data that Morgan talked about, I'll be estimating population parameters, so those will be the population size, population density, survival rates, and population growth rates. And I'll mainly be using program ARC to estimate those things, but I'll also be using a package within the software program R where I can actually use some of that same spatial data and look at spatially explicit density estimates. I'll also be using the blood samples, and I'll take those and extract DNA. I'll perform PCR to amplify 15 different microsatellite loci, and then I'll use those genotypes to estimate the effective population size, which I'll give you a definition for in the next slide. So, as promised, before I present a couple of preliminary results, I'm going to give you these definitions so you know what they are. The census population size is simply the number of individuals that are out there in the population or the study area. And ideally, if you could go out and count every single snake, that would be great to get this, but because you can't do that, they're pretty hard to find, especially these last few weeks. We have to use that market capture data, and that can allow us to actually use detection rates and estimate the number of individuals. And in this case, we're interested in adult snakes. The effective population size is a roughly estimates the number of adults that are breeding within the population. And so that's important because that's what determines the amount of genetic diversity. Also, in the wild, the effective population size is always going to be smaller than a census size, and that's true across all types of wildlife. And a lot of times, effective population size is actually much smaller. So, some preliminary results. Using last year's market capture data, because that was the best year of data collection so far, we used 77 different individuals and 190 captures of those individuals, which were all adults, to to estimate that there is 105 adults in the population and within the study area. And this accounts for some uncertainty around the estimate. So we have a 95% confidence interval that there are 86 to 161 adults. For the effective population size, we have 46 individuals that went into the genotype, or that we genotyped at this point 9 microcentilli loci. And the estimate for effective population size is 56 individuals with a confidence interval of 36 to 103. So for the rest of the summer, Morgan and Arin are going to complete their GIS analyses that they talked about. I'll be using this year's data to get another estimate of the current population size and population density. And then in the future, as I finish up my master's project, I'm going to continue my lab work, so I'll genotype those other 6 microsatellites, and that will improve that estimate of effective population size, or it should anyway. I'm also going to compare the effective and census population size here at Pierce with a couple of other populations, and one for sure that I'll be looking at is in Cass County, Michigan. And then I'll also use all the data from 2011 through next summer to estimate survival rates and population growth rates of Massasoits. And with that, I will take any questions.
So a couple of quick ones. Don't you think that maybe it was just the polygon size of your GIS underlayer? I mean, in other words, your snakes are probably spot on in the lake, but you know, if your resolution for your underlayer is X and you're, you know.
Yeah, it was from a really, really, really big, giant raster file, so
yeah.
we're just going to do our best to go back and reassign the values to coordinate to match the adjacent wetland.
Right. Anyway, so that was one quick one. And then another quick one was about the effective population size. I was curious about